So, imagine you run 5 miles a day, right? You maintain perfect blood pressure. You have this flawless family health history. You basically feel completely invincible.
Right, like then nothing could go wrong.
Exactly. But then, purely by chance, a single drop of your saliva reveals this genetic time bomb just hidden in your cells. A bomb that's quietly ticking, but, well, one you can actually defuse before it ever goes off.
And that right there, that scenario represents, honestly, the ultimate blind spot in modern medicine. Because for decades, our entire healthcare framework has been built on this, like, reactive, wait until you're sick model.
Yeah, you don't go in until something's wrong.
Exactly. You wait for the symptom, you wait for the tumor, you wait for the heart attack, and only then do you try to fix it. So finding those hidden risks proactively, that's a monumental paradigm shift.
Which brings us to the core mission of today's deep dive. We're looking into the Gen4ME study. That stands for Genomic Medicine for Everyone, and it was rolled out by Providence Health.
Yeah, and the scale of this is just incredible.
It really is.
Yeah.
That's what caught my eye initially, because this isn't some tiny pilot program hidden away in an ivory tower research university. Providence is this massive, decentralized community health system. I mean, we're talking over 50 hospitals, 1,000 clinics spanning across 7 states.
The huge network.
Right. And their goal was to figure out how to integrate whole genome sequencing, or WGS, into routine, everyday healthcare.
But before we get into the heavy logistics of that, I think we need to clarify what whole genome sequencing actually does, compared to a standard genetic test you'd get at your doctor's office today.
Oh, for sure. I usually think of traditional genetic testing like using the index of a book to check a few specific pages. Like if your mom had breast cancer, the doctor flips to the index, finds the BRCA gene, and only reads that one page to see if you have it too.
That's a great way to look at it.
But WGS, whole genome sequencing, is like reading the entire encyclopedia of your biological makeup, cover to cover, every single letter, completely regardless of your family history.
Right. And in this deep dive, we're going to explore how Providence Health deployed that massive encyclopedia scan across the general population, specifically to find hidden risks that standard medical screening it simply cannot see.
So, to you listening right now, have you ever wondered what genetic secrets are hiding in your DNA? Things completely disconnected from what you know about your family tree. Let's get into the mechanics of how the Gen4ME study actually pulled this off, because honestly, the sheer logistics of screening a population like this are staggering.
They really are. I mean, the recruitment and e-consent phase alone required a massive digital infrastructure. They initially reached out to over 30,800 potential participants.
Wow, over 30,000.
Yeah, a huge net. And from that initial outreach, 2,716 people formally consented to the study.
Okay, so that's, what, an 8.8% enrollment rate?
Exactly, 8.8%. And by the end of the entire pipeline, 2,017 people successfully had their sequencing completed and actually received a clinical results report.
I noticed they used a multilingual e-consent platform for this, right?
Yeah.
Like in both English and Spanish.
They did, yes.
Which means no paper forms, no mandatory in-person clinic visits, just to sign a clipboard. I have to assume removing that physical friction is like the only way a health network can actually scale a study this big.
Oh, absolutely. Digital onboarding is essential for scale, but, digital consent still requires a physical biological sample. So the participants received an origin saliva DNA kit in the mail. We can just call it a spit kit.
Spit kit, got it.
Yeah, so the participant spits in a tube at home and mails it back. But occasionally a saliva sample fails to yield enough high-quality DNA.
Oh, really? It just doesn't work?
Yeah, it happens. And when that happened, the study had a backup protocol where the participant provided a blood sample instead. But eventually, all of these samples converged at the Providence Molecular Genomics lab.
So I'm trying to picture what happens next. The lab receives thousands of tubes of saliva. How do they physically turn spit into a digital encyclopedia?
Well, they utilized this incredibly sophisticated piece of bio-IT infrastructure. It's called the Illumina Dragon platform.
Dragon, like the fire-breathing kind.
Spelled a bit differently, but yeah. It stands for Dynamic Read Analysis for Genomics. What it does, it takes the raw chemical data extracted from the cells in your saliva, and it translates it into a digital sequence. But they aren't just reading the code like a novel, They're actively hunting for very specific structural errors.
Right, and I read the study's breakdown of those errors, and honestly, the terminology is a bit dense.
It's very technical, yeah.
They were looking for SNVs, indels, and CNVs. Now, I know SNV stands for single nucleotide variant, but I don't really grasp what that fatally does to my body? Like, how does one tiny variant cause a massive disease?
It's a great question. Let's follow the mechanism all the way down. So your DNA is basically a recipe book for building proteins, right? An SNV, that single nucleotide variant, is a single letter typo in that recipe.
Just one letter.
Just one. So if the recipe calls for sugar and the typo changes it to salt, the final product is completely ruined. In your body, that one wrong letter means a cell builds a protein that's folded the wrong way.
Oh.
Yeah. Suddenly that protein can't do its job, like, say, suppressing a tumor, and so a cancer cell's allowed to grow.
Okay, that makes perfect sense. So an SNV is a typo. What about indels?
Right, so indels stands for insertions or deletions. This is where extra letters are just shoved into the recipe, or crucial letters are missing entirely. It shifts the entire reading frame of the DNA, which usually results in a completely non-functional protein.
Okay, and the last one, CNVs, copy number variants.
So if an SNV is a single typo, a CNV is a case where whole paragraphs or even entire pages of the encyclopedia are either duplicated sometimes multiple times over, or they're ripped out completely.
Jeez, so chunks are just gone or copied.
Exactly. The sheer volume of the genetic material is altered.
Even just mapping those errors for one person sounds exhausting.
Sure.
But doing it for over 2,000 people, I mean, that means analyzing an ocean of data. There's no way a team of human doctors is just sitting in a room reading all that.
No, absolutely not. They relied heavily on AI to do the heavy lifting here. They used a system called ACE, that's the Automated Variant Classification Engine, developed by a company called Genomics.
Okay.
ACE is programmed with the 2015 guidelines from the American College of Medical Genetics and Genomics, or ACMG. It basically scans millions of variants and scores them based on their potential danger.
And the study actually categorized these AI findings into 3 distinct buckets, right? I think a traffic light analogy is probably the best image memory tool for us to use here.
It's like that analogy.
Yeah. So let's start with the red light. If the AI found a PLP variant, meaning pathogenic or likely pathogenic, that is a red light, it means this variant is actionable, it's high risk, and these are the results that were actively returned to the patient.
Right, the things they need to know about immediately.
Exactly. Then you have the yellow light.
Yeah, the yellow light variants are what we call VUS, or variants of unknown significance. This happens when the AI spots an anomaly, but the global medical community simply doesn't have enough data yet to confidently say whether it causes disease or if it's totally harmless.
So what do they do with those?
Well, yellow light results were deliberately not returned to the patients.
Right.
Because telling a patient they have a mystery mutation, it usually just causes severe psychological distress and can lead to unnecessary invasive medical procedures.
That makes a lot of sense. You don't want to panic people over nothing. And then finally, the green light variants, the BLB, which stands for benign or likely benign.
Exact.
These are just harmless variations in your code. Like
Yeah.
they might dictate the color of your eyes or some weird quirk in your metabolism, but they don't make you sick.
Right. They're just what makes you you. So let's do a quick mini recap of what we've covered in this first segment, just to lock it in. We are looking at the foundational mechanics of Geno4ME. First, they mailed spit kits to thousands of people using a really frictionless digital consent platform. Second, they used the Illumina Dragon platform to translate the chemical DNA into digital code, actively looking for structural errors like SNVs and CNVs.
Got it.
And third, they used the ACE AI to sort through that massive data pool, filtering the results to only alert patients to the red light, highly actionable pathogenic variants.
So to the listener, think about this.
Yeah.
Would you be willing to spit in a tube and mail it off if you knew an artificial intelligence was going to scan the deepest blueprints of your body for life-altering red flags? It's a heavy question. But for the 2017 people who went through this process, the results were really eye-opening. Let's look at the inherited disease discovery.
Yeah, so researchers focused the AI on a very specific genetic panel containing 78 inherited disease genes.
Now, I'm assuming they didn't just throw darts at a board to pick the 78, right? Is there like a master list of the worst offenders they work from?
Oh, the selection was highly regulated and entirely evidence-based. So 59 of those genes are designated as actionable secondary findings by the ACMG, that's the American College of Medical Genetics and Genomics.
Right.
And the remaining 18 genes were added based on strict criteria from the NCCN, which is the National Comprehensive Cancer Network. They were really looking for diseases where early detection actually changes medical management.
Okay, so here is the core data they found. Out of everyone who received a report, 7.8%, which equates to 158 people, had at least one red light pathogenic variant.
Yep. 158 people.
And across those 158 people, the AI flagged 163 total pathogenic variants. But here's the wild part. A striking 55.8% of these positive findings were in cancer-associated genes.
Yeah, but honestly, the most staggering statistic in this entire study is this. Of the participants who received a positive life-altering red light finding, 52% of them had zero self-reported personal or family history of that disease.
Wait, I need to stop you there because 52% is massive. That's more than half. Currently, if I go to my doctor and ask for a genetic test for cancer risk, my insurance will usually deny it unless I can prove a strong family history. Doesn't this mean our entire healthcare system is basically flying blind? Like actively ignoring half the people carrying these time bombs?
It does. It exposes a massive flaw in how we gatekeep genetic testing. Family history is actually a terrible filter in the modern era.
Really? Why is that?
Well, families are much smaller now than they were 50 years ago, which limits the data pool. Plus, people lose touch with their relatives. Or, very commonly, previous generations passed away from things like heart attacks, accidents, or, other illnesses before a genetic cancer ever even had the chance to develop.
Oh, that makes a lot of sense.
Right. So relying strictly on the family tree means abandoning that 52%.
And looking at the specific cases they found really highlights how dangerous it is to fly blind, because 3 participants actually had double mutations.
Yes, double mutation.
Like, one person had a pathogenic variant in both BRCA2 and CHK2, another had mutations in ATM and LDLR, and a third had mutations in ATP7B and KCNH2.
And a double mutation fundamentally alters a patient's risk profile because your DNA has multiple redundant defense systems, right? So a mutation in one gene is like a security camera going down. But a double mutation is like the camera going down and the security guard falling asleep at the same time.
Wow.
Yeah, it requires complex, multidisciplinary medical intervention to manage.
And the risk levels for these genes vary wildly, don't they? Like high-risk genes like BRCA1 and BRCA2, they carry a lifetime cancer risk of over 40%.
Over 40%, yes.
The panel also looked for MLH1 and MSA6, which are associated with Lynch syndrome and highly aggressive colorectal cancers.
Right, those are the high-risk ones. But we also have moderate-risk genes, and this is where the medical reality becomes deeply, deeply complicated. Well, the most common moderate risk findings in the entire Genoform E study were in a gene called MU2IH, which was found in 30 people, and a gene called Chi2 found in 18 people. Specifically, of those 18 people with a Chi2 mutation, 7 of them had a specific variant known as I157T.
And the study points out something pretty wild that happened with those specific MU2IH and Chi-T variants while the trial was actually ongoing. The NCCN guidelines actually shifted underneath the researchers' feet, didn't they?
They did. Medical science is constantly evolving as global data aggregates. So initially, the NCCN guidelines stated that if you had the MU2IH mutation or the Chi-Chi-P2 I157T variant, you required enhanced medical screening. For example, you might be told to schedule an early urgent colonoscopy.
Okay, so that was the initial rule.
Right. But then newer global data came in, and the NCCN updated their guidelines. They actually downgraded those exact variants to just carrier risk.
Meaning what?
Meaning the medical recommendation shifted from get a colonoscopy right now to you actually don't need any extra screening at all.
That's crazy. This psychological whiplash of that would be devastating for a patient.
Exactly. Imagine being the patient sitting in that clinic. On a Tuesday, your doctor looks you in the eye and tells you that your DNA requires urgent cancer screening. You go home, you tell your family, you stress out about the procedure.
And then
a month later, your doctor calls back and says, actually, the global guidelines just updated. You're fine. Cancel the screening.
it creates this massive risk of over-screening patients.
Yeah.
And I imagine it places an intense burden on health systems too, because they have to constantly monitor databases and recontact patients as the science evolves.
It's a huge logistical and ethical challenge. Let's do our second mini recap here to lock in these concepts. First, population screening found that roughly 1 in 13 people have a serious genetic risk.
Right.
Second, 52% of those at risk would have never qualified for standard genetic testing because their family tree looked perfectly healthy. And third, interpreting this data is a moving target. Guidelines for genes like MUTYH and Chi2 can shift dramatically in real time.
So, think about this. How does this change your trust in standard medical advice? Knowing that the textbook used to interpret your very DNA can literally be rewritten overnight. It's deeply unsettling, but I think it also proves just how fast our understanding of the human body is accelerating.
It really is.
Now, let's pivot away from inherited cancer risks to something vastly more common. Section 3. Pharmacogenomics, or PGX.
PGX, yes. This is the study of how your specific genetic code dictates your body's response to pharmaceutical drugs.
I used to think of drug metabolism with, like, a terrible car analogy, like putting premium gas in a diesel engine. But it's actually much more mechanical than that.
Oh!
Yeah, think of your liver like a custom-built microscopic recycling center. The enzymes in your liver are the shredders. If your DNA gave you a massive industrial-sized shredder, you might destroy a medication before it ever reaches your bloodstream to actually help you.
Right, you metabolize it too fast.
Exactly. But if your DNA gave you a tiny wheat shredder, that same medication piles up in your liver, it becomes highly toxic and it causes severe side effects.
That is a highly accurate way to look at it, actually. And the Geno4ME study used a PGX panel targeting 4 specific genes that control those shredders.
Oh, were they?
CYP2C19, CYP2C9, VKORC1, and CYP4F2. The CYP genes, we pronounce it CYP, are responsible for producing cytochrome P450 enzymes. And depending on the variants in those genes, you are either an ultra-rapid metabolizer, a normal metabolizer, or a poor metabolizer.
And they focused these 4 genes on 7 commonly prescribed everyday drugs. We are talking about antidepressants, specifically SSRIs like Celexa and Lexapro.
Wow.
We're talking about PPIs, which are heartburn medications like Prevacid, Prilosec, and Protonix, and incredibly important blood thinners like Plavix and warfarin.
Yeah, and these drugs were selected based on highly rigorous guidelines from CPIC, the Clinical Pharmacogenetics Implementation Consortium, as well as official FDA drug labeling. Because, I mean, the margin for error on a blood thinner like warfarin is razor thin.
Really thin.
If you are a poor metabolizer, a standard dose could cause massive internal bleeding.
Wow. And the results of this PGX panel are something every single person needs to hear. 100% of the people tested have at least one PGX variant that affects their medical care.
100%.
Every single human being has a unique metabolic fingerprint. But more specifically, 14.6% of the participants had a potentially actionable result for just those 7 specific drugs.
And this is where genomic sequencing directly impacts daily life. Because of these findings, 39 people, which represents 13.3% of those who received a PGX report, had a pharmacist actively step in and alter their current ongoing therapy.
That is incredible. That means pharmacists looked at the DNA, realized the patient's liver enzymes were either shredding the drug too fast or letting it build up toxically, and they made a change.
Right then and there.
They modified dosages, completely stopped certain medications, or switched the patient to an entirely different therapy purely based on reading their CYP and VKRC1 genes.
So think about the heartburn medication or the antidepressant you or a loved one might take every single morning. What if the reason you still feel awful or the reason you have lingering brain fog isn't because of your diet or your stress levels?
Wow.
What if your genetic code literally prevents your liver from processing that specific pill?
It completely reframes how you look at a pharmacy counter, doesn't it?
Yeah,
A pill is only as good as the enzymes processing it.
it really is. So let's do a quick recap of Section 3. First, pharmacogenomics proves that 100% of us have genetic quirks in how our liver processes drugs.
Yeah.
Second, by testing just 4 genes against 7 common drugs, 13.3% of patients had their daily prescriptions optimized by a pharmacist, preventing toxic side effects or ineffective treatments.
We've talked a lot about the incredible potential of this data, right? Catching cancer early, optimizing daily meds. But honestly, none of that matters if this technology only reaches a privileged few.
That's the real challenge.
Exactly. So how did Providence actually ensure equitable access to these tests? That brings us to Section 4, equity, access, and the follow-through.
Well, historically, genomic research has an abysmal track record with diversity. I mean, data sets heavily skew towards white populations of European descent, which limits the accuracy of the AI for everyone else.
Right.
Geno4ME actively set out to close this gap by comparing 2 recruitment methods. They had clinic invitation versus population outreach.
So clinic invitation is exactly what it sounds like, right? Asking people to join the study while they're physically sitting in the doctor's office.
Yes.
Whereas population outreach was this broad digital net, utilizing email, physical mail, and text messaging in both English and Spanish, targeting people who hadn't necessarily scheduled a doctor's visit.
And the demographic results of those 2 methods were drastically different. The digital population outreach was vastly more effective for ensuring diversity. 57.2% of the outreach group identified as racial or ethnic minorities.
57.2%.
Yes. Compare that to the clinic invites, where only 9.2% identified as minorities.
Wow. So reaching people digitally clearly works better for initial consent. But the study highlighted a fascinating behavioral hurdle when it came time to actually provide the saliva sample.
The spit kit.
Right. White participants had an 80% return rate for the physical spit kits, but Hispanic participants had a 69% return rate. I'm trying to figure out why there is an 11% drop-off there when both groups already consented digitally.
It really comes down to the deep friction of physical reality versus digital ease. Tapping, I agree, on a smartphone is fundamentally different from collecting your own biological material and handing it over to a massive medical institution.
That's fair.
There are deep layers of historical mistrust regarding how minority DNA has been used in the past. Combine that with potential language barriers regarding the physical postal instructions. Like, even if the digital consent was in Spanish, the physical mail system can be daunting and feel invasive.
It really emphasizes that technology can bridge the communication gap, but building institutional trust requires so much more work.
It does. However, for the patients who did complete the process and received a red light finding, the institutional follow-through was phenomenal. Remember those 158 people with inherited disease risks?
Yeah, from the first section.
Well, 154 of them were successfully referred to an external partner called Genomics for telehealth genetic counseling.
And an incredible 88% of them actually completed that telehealth appointment. I mean, that is a massive compliance rate.
It's unheard of. Usually, making a patient schedule a separate specialist appointment results in huge drop-offs. Telehealth completely removed that barrier.
And crucially, during the window of this study, there was not a single reported insurance denial for the recommended care changes that stemmed from these genetic findings.
Zero denials.
Zero. All right, time for our final subtopic summary. Broad digital multilingual outreach is essential to diversify genetic testing, achieving a 57.2% minority participation rate in the outreach group. But behavioral friction still exists in mailing physical samples, requiring better trust building. And finally, pairing results with telehealth counseling achieved an 88% compliance rate, proving that patients will take action if you make it accessible.
So think about your own life. What physical, cultural, or even logistical barriers would make you hesitate to participate in a remote medical study? Is it privacy concerns, distrust of the lab, the friction of the mail system? Understanding those hesitations is literally the key to the future of public health.
If we pull all of this together, the Gen4ME study proves something vital. It proves that population-scale whole genome sequencing isn't science fiction, and it doesn't just belong in elite research labs. It can be successfully, equitably, and safely integrated into a massive decentralized community healthcare system right now.
We have the AI to read the code, we have the platforms to consent the patients, and we have the telehealth infrastructure to counsel them. The tools are here.
Let's circle back to where we started. You walk into your doctor's office, you feel healthy. The standard tests are clear. But your genome sequencing reveals a hidden risk. You catch it early. You defuse the time bomb. You are safe.
It's a great outcome.
It is. But I want to leave you with a final provocative thought to mull over. If your whole genome is sequenced today, that digital encyclopedia is yours for life. The sequence of your letters doesn't change. But as we saw with the shifting cancer guidelines for genes like MUTYH and CHK2, the science of interpreting those letters is constantly updating.
Always changing.
So 10 or 20 years from now, whose responsibility is it to call you and say, hey, remember that variant we told you was harmless back in 2026? We were wrong. The science changed. We need you to come in.
That's the million-dollar question.
Is it your primary care doctor's job to keep checking a file you made 20 years ago? Is it an AI's job to constantly scan global medical databases and send a push notification to your phone? Or does the responsibility to constantly check for updates ultimately fall on you?
Something to really think about.
Keep questioning the information around you, and we'll see you on the next deep dive.